Hello friends, let's start the new video lecture of Pramesh notes. In the previous video we learned about spermatogenesis as a part of gametogenesis. The process of oogenesis is quite similar. The first step of oogenesis is multiplication phase, second one is growth phase, and third one is maturation phase. The oogenesis is process longer as compared to spermatogenesis. In spermatogenesis, primary spermatocytes is divided by meiosis 1, forming two secondary spermatocytes, whereas in oogenesis, primary oocytes is divided by meiosis 1, forming secondary oocytes and one polar body. In the multiplication phase, germinal epithelial cells in ovary's outermost layer undergoes mitotic cell division to form oogenia, 4 in number, represented by 2n. The oogenia are tiny diploid germ cells that pass through mitotic divisions and originate the primary oocytes in the growth phase. In the growth phase, oogenia converts into primary oocytes. Here the development of DNA, RNA, ribosome and Golgi bodies happens. The growth phase is longest phase in oogenesis as it converts into a large primary oocyte surrounded by a layer of granulosa cells to form the primary follicles. In maturation phase, primary oocyte is converted into secondary oocyte through meiosis I cell division, and secondary oocytes is converted into ova or egg through meiosis II cell division. In conclusion, from oogenium, one ovum and three polar bodies are generated. Polar bodies don't reproduce and so degenerate quickly. Just when the ovum is formed, it waits for fertilization process and sperm to enter. Let's learn about the structure of eggs and its classification on different bases. On the basis of amount of yolk, eggs are classified into microlecithal, mesolecithal, and macrolecithal. In microlecithal, there is little amount of yolk. It occurs in starfish, amphibious, eutherian mammals like rabbit and humans. In mesolecithal, there is moderate amount of yolk. It occurs in amphibians like frogs. In macrolecithal, there is large amount of yolk. It occurs in arthropods, fishes, reptiles, birds, and monotremata, protherian mammals. On the basis of distribution of yolk, eggs are classified as isolecithal, telolecithal, centrolecithal, heterolecithal, and myolecithal. In isolecithal, yolk is uniformly distributed in cytoplasm like in protochoroids and echinoderms. In telolecithal, yolk is concentrated toward vegetal pole. The nucleus and major part of cytoplasm is displaced to animal pole. It occurs in frogs, reptiles, apes, and fishes. It exists in mesolecithal and macrolecithal eggs. In centrolecithal, yolk is concentrated in center of ovum, surrounded by cytoplasm. It occurs in arthropods as the macrolecithal eggs. In heterolecithal, yolk is unevenly distributed. In myolecithal or discoid, whole egg is occupied by yolk except a small disc of active ooplasm. It occurs in birds and reptiles. Let's learn about slioidic eggs. Slioidic eggs are those eggs on which cells are present. These kinds of eggs occurs in reptiles and birds to stay insulated from external environment. 94% of cell is made of calcium carbonate. Whenever we see eggs without cell, we call it as aslioidic cells. This much in this video. See you again with the next topic of embryology, that is fertilization.